Okay, so here I am standing at my desk, and I'm going to talk to all of you about implementing 21 molecular algorithms using reprogrammable DNA self-assembly. This work was done in collaboration with Dave Doty, Eric Winfrey, Cameron Mirho, Joy Hoy, Felix Zhao, and Peng Yin. And we did the experiments primarily while Dave and I were at Caltech, and a lot of analysis happened later on when I moved to INRIA and Maynooth University, and Dave went to UC Davis. All right, so one piece of background here is that we're going to be talking about algorithmic self-assembly, or implementing programs via self-assembly. And so one piece of background that's important is to just mention about two technologies. One is DNA origami, that I'm sure many of you know about, where you have a long scaffold strand that gets pinned or stapled together by small staples to make some designed shape. So you design your little staple strands so they'll pull together this long strand to raster into some desired shape, like, for example, this rectangle with these smiley faces. Another technology from Wei et al. In 2012 is to use single-stranded tiles, where you have individual short strands, here about 42 bases long for each one, and you design their sort of sticky end interactions such that they come together and make a square in a square lattice. So, for example, this square or rectangle shown here, or you could leave some tiles out and get these letters of the alphabet. So when we make structures like that, one way to think about it is that for each pixel in the shape that we're building, we're telling that pixel exactly what DNA sequence it should get. So in the tile-based systems, for each pixel, you've got a tile, and that tile, or those 42 bases, uniquely represent that pixel, all right? And so then in the your hope is that in the equilibrium distribution, you find these shapes and nothing else. And for origami, that's certainly often the case. So, however, imagine we could so this is sort of analogous to having an architect who is a control freak, and they tell every brick in the building exactly where it should be. They give a unique address to each brick in the building they're designing. And it would sort of be interesting from our point of view to have smarter molecules that decide where to go for themselves. So where the molecules execute some logic or a computer program to decide where to go for themselves. So that might seem like a hand-wavy statement, especially since I use the word smart, which is a vague term. So now let me make it extremely concrete. I'll tell you exactly what I mean by smart or by logic in self-assembly. So imagine I'm growing some structure from left to right across my screen, and there's a crystal that's growing, and basically I have these monomers that can stick to the growing lattice and make it bigger, but when they stick, they're going to have a very special rule. They're going to stick to that lattice if and only if they match on two colors. So here we've got gold matches gold, but brown does not match gold. So that molecule, which diffused up to that surface right there, to that growing frontier, it just diffuses away. Here's another one that diffuses by, but it matches on neither color, so it just falls away. And this third tile, now this third molecule, it matches on one side, but not the other, so it falls away. And then finally, we have the perfect molecule that matches brown, gold, and so it sticks there. If you notice here, I wrote down symbols 0 and 1 on my color. So brown is 0 and gold is 1. And in fact, if I think of the two left-hand sides of the tile as inputs and the top right-hand side as an output, these tiles are executing OR logic. 0, 0 goes to 0, 0, 1 goes to 1, 1, 0 goes to 1, 1, 1 goes to 1. And I could have put any gate I want in there, any two input, one output gate. And indeed, I could make my molecules twice as smart now by making use of this sticky patch down here, this other output that I've not made use of yet. And I could put some colors there. I could put any pattern of brown and gold that I want. So let's just put brown, 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 gold, which corresponds to AND logic. So now I'm going to think of my four tiles, my four molecules, as encoding an OR gate on the top and an AND gate on the bottom. And so I could write it symbolically or using this diagram on the right-hand side. And then there's no need to write out all these zeros and ones because we know what these little pictures mean. So I could just write it like this. So there's an OR gate and an AND gate. All right, okay. So from now on, this blue thing in a circle, I'll just call that a gate. It's got 2 inputs and 2 outputs. And as this crystal is growing from left to right, it's going to execute logic as more and more tiles come and fill in the crystal. So instead of thinking about the crystal, let me abstract away now and just think about gates for a minute or two. So we're actually programming a circuit model here. Okay, so I could imagine some sort of Boolean circuit model, but where the wires are local wires. All right, so I've got gates as shown by these circles, and then gate number 3 is wired to gate 2 and 4, both its inputs and outputs, and each gate at each position could be any function I want. Here at position 3 I have OR and at position 6 I have the NOT of the top bit and the NAND of both bits. Okay, so I could put whatever I want at each position. I could even have randomized circuits where at a given position I could have several gates with a probability associated to each gate, and the probability should sum to 1. This would be useful for writing randomized algorithms later on. And so then, okay, how are we going to program this thing? Well, the programmer just needs to specify one of these layers. And that's all they do, okay? So for a 6-bit system like this, the programmer just specifies 5 proper gates and then a kind of a special case for the top and the bottom with a single input and a single output. And then a user might give an input. So this is a 6-bit system, so it just gets 6 bits of input. And then the way the system computes is just copies and pastes the same layer forever, okay? So I've now defined a circuit model of a certain kind. It's a 6-bit version of it, but you could imagine this for any even n. All right. Also, when I'm copy pasting, if at a given position I've got a randomized gate, so there's 2 or more gates there, then you would sort of pick one of those randomly according to the probabilities that were given by the programmer. Okay, let's see an example. So I'm just going to use that OR AND example I used before, and I'm going to put OR AND gates at each position in the layer on the left. So that's what the programmer does. The system executes by taking some input bits and then copy pasting that layer forever. All right. So let's give an input. So in this case, I'm going to give 5 zeros and a 1. And instead of tracing through this circuit diagram, I'm going to draw a little picture on the top where the 1s are denoted by a yellow circle, just like I had yellow on my tiles earlier. And the zeros are denoted by brown. And you can see in this case that what this circuit does is it shuffles the 1 bit to the top and the zeros to the bottom. And in fact, if you give any 6-bit input, it will do that. It will shuffle the 1s to the top and the zeros to the bottom. So we call this bit sorting. All right, so it's sorting the bits. And in our work, we implement this using DNA tiles by having a lattice that grows from left to right, as I was showing schematically earlier, but each of those little square tiles is going to be encoded using DNA strands. And then our bits are going to be represented by DNA strands that streptavidin proteins stick to, and then we can see them here on these AFM images. So each of those little circles is about 5 nanometers across. All right, so here's another quick example. This is a really boring circuit. It just copies bits to the right. So you've got the programmer, who is one of the least interesting programmers ever, has just made this circuit on the left that passes the bits through. And so any input just gets copied to the right. Okay, fine. Why did I show you that example? Well, it allows me to then show you a very simple randomized circuit where at each position we're going to have two gates, the little sorting gate with the OR and the AND we saw earlier, and then this pass-through copy gate. Each of them gets probability a half. So what's going to happen now? Well, we give some 6-bit inputs, and at each step you either sort or copy each time with probability 1/2. Roughly speaking. So here we've copied for a while, and then we sort, and then we copy, and then we sort. And then we also implemented that circuit using DNA And you can see here what amounts to, under our AFM, a tape recording of this randomized computation. So you have these events which are happening very quickly and at the nanoscale, and we don't have a way to image them as they're happening on a single molecule level, but we are able to image this tape recording of those randomized events, which I think is really cool. All right, so I showed you there 3 circuits, and at the beginning of this project, we knew that, okay, we've got a circuit model here, and we'd like to build it using DNA. But as theoretical computer scientists, we also knew that, well, the longer we stare at this circuit model, I mean like over the course of weeks and months, the more algorithms we're going to figure out how to run on this thing. So the problem is if we buy all the DNA strands now for those 3 algorithms, then we're going to want to buy more later on. So instead of taking that approach, we just noticed that, well, there are very many circuits out there, but in fact, they can all be like billions of them. But they could be built using a small number of gates, right? So with 1288 gates, you could implement any 6-bit circuit in this model, all right? So there's a finite number of possibilities for each position, and then there's only certain ways to wire them up. Okay, so that set of 1288 gates, if I had that sitting in my drawer under my lab bench, then I could, wire up any circuit I want. Just take out the gates that you want to use that day. Okay, cool. All right, let's implement that. So instead of implementing one algorithm, let's just build all the gates, and then we can wire them up as we like and implement any algorithm. So this is kind of like the idea of universality in computer science. The fact that my laptop can run MS Teams and my presentation and do lots of other things. So we want to use this idea of being able to run any algorithm of a certain kind. Of course, ours is only of the 6-bit kind in this IBC model, we called it. Okay, so how are we going to do that? Let's do that. All right. I've already shown you that you can take 4 tiles that map 2 bits to 2 bits and consider them to be a gate of a certain form. And so here we're just going to go in the other direction. We're starting from the set of all gates, and we want to map each gate to 4 tiles. So we just do that as shown here. So a gate has a truth table, and each truth table line becomes a tile. So for example, 0, 1 maps to 1, 0 becomes the tile, 0, 1 maps to 1, 0. I hope folks can see my mouse. Okay, so then the circuit computation, which happens layer by layer, will correspond to growing an assembly starting from some seed structure with an input bit string, and then it growing sort of tile by tile, which ultimately looks like layer by layer after it's done. And when you do that transformation, interestingly, the number of gates, which was 1288, goes down when you turn it into tiles, down to just 89. And that's because each tile, like this tile, can appear in many gates. So that particular line in the truth table shows up in many gates, this gate and some other ones. And so when you crunch the numbers, you just need 89 tile types. There's an odd number here because we're doing something funny at the border. Okay, let's build these out of DNA. We're going to use single-stranded tiles that came from this paper by Yin et al. In 2008, and actually the same kind of tiles that showed up in this paper by Wei et al. In 2012 that I showed you earlier. All right, so we're going to think of those as our little squares. If you look in the lattice here, they bind each tile, like this light blue one in the center, binds to 4 tiles, orange, green, brown, and pink. All right, okay, so let's do that. Let's take the square tiles and encode them as DNA strands. Okay, one issue here is that if you do that directly, it turns out that you get some errors in the assembly process that are a little too large for what we'd like. So we didn't do a careful study of this for these kinds of tiles, but maybe you could think like something like 3% or 5% of an error rate could be within an order of magnitude perhaps. And so your computer would be pretty bad if it was making mistakes 3% of the time. So what we did was we used a theoretical technique called proofreading, by Winfrey and Beck-Bolotov. So one of my collaborators is Eric Winfrey here. And what you do in this technique is you take each tile and you replace it with a block of tiles. So in this case, we took each tile and replaced it with a 2x2 block of tiles. So there's a little bit of redundancy here, is the quick intuition as to why this works. So if there's going to be an error in the system, it actually forces a second error. And if your error rate is low enough, that kind of quadratically decreases the error overall. So now what happens is each of our single tiles here gets replaced by 4 tiles. So we had a gate, it mapped to the 4 tiles on the left, and they get mapped to 16 DNA tiles, 16 DNA strands on the right that have the same functionality. And so instead of growing this lattice shown on the bottom left here from this seed, we're going to have some sort of DNA structure that's going to self-assemble. Starting from some seed structure on the left, we'll use a DNA origami for that. It'll encode the input bits. And then we'll have tiles attached and grow something to the right. A few technical points here. We're going to have the bottom of this structure fold around and bind to the top. So the whole thing folds around into a tube. So it looks like a flat ribbon here on my slide on the bottom right, but it's actually going to be a tube. The bottom binds to the top. And that allows for all tiles to bind in the same way. There's no special conditions at the border that made our life easier. We're also going to hard code the rows, meaning that if you take a column, there's no tile types repeated along the column. There's unique sequences along the column. This stops the tubes sort of folding up too early. Like what you don't want is these tiles, say along U2 here, binding to U6, right, and making a smaller tube. So we don't repeat tile types on the columns. We're going to label the one, so there's some tiles with yellow patches on them here that denote they encode 1 bits as opposed to zeros. And we're going to label those with biotin, so those DNA strands are going to have a biotin internally on them, and that will allow us to flow in streptavidin protein, which binds there, and then we can see them on AFM. Okay, so when we do that transformation, our 1288 gates became 89 tiles in the previous slide, but it got blown up by a factor of 4 here, which is 356, and then due to something special at the border, that goes down to 355, so we end up with 355 DNA strands. So the previous most complicated algorithmic self-assembly system implemented was around 20, 22, something like that, DNA strands. So this is a factor of 15 scale up in complexity for design, which made this task challenging, DNA sequence design. And so what we want here is sort of encapsulated by the figure on the bottom left. We'd like correct attachments to happen, but we'd like incorrect attachments not to happen. Remember this rule I said earlier on, where we wanted we want a tile to bind if it matches on both colors, like shown here with the green tick, and we do not want the tile to bind if one of the colors mismatches. So if you just choose random sequences for your single-stranded tiles, then you'll get tiles binding with you get errors all the time. Basically, you get a massive overlap in the strength of binding for binding with one correct side versus 2 correct sides. And so we spent quite some months working on DNA sequence design to separate these 2 histograms as shown on the right-hand side, so that binding with one side in blue is much weaker than binding with 2 sides, which is shown in right in units of minus kcal per mole. Okay. There's a whole story here that I don't have time to talk about today, but this was a really interesting part of the project to figure out the right set of criteria to use here, and there's still a lot of open questions around that. All right, we need a seed. For that, we use a DNA origami, which I mentioned on my second slide. So we're going to have the DNA origami that folds into, so the design so it folds into a barrel, and it's going to act as a seed. It's going to have 2 features that make it really nice. One is we're going to write some digits on the inside of the barrel so that when we put it down on mica, we can see those digits. They're written with biotin modifications on the DNA, and then we flow in streptavidin and we can read these numbers. So it'll allow us to mix our samples together and have different barcodes for different samples. The other feature that's nice for this barrel is that we have the ability to unzip it and make it into a flat rectangle. So after it's formed, we can send in some strands that rip open one of the helices, or two adjacent helices are pulled apart, and then it turns into a rectangle. This will be useful for imaging. All right, let's look at our results. Okay, in order to understand our results, I'm going to explain what an experiment looks like. Well, the first part of an experiment uses your imagination. You come up with a circuit, so it might be sorting, it might be something else. I'll show a few examples soon. But you go to the whiteboard and you figure out a circuit that has a cool behavior that you'd like to see implemented. And now we're at the position now where we've designed our 355 DNA strands, and they're sitting in the fridge. So I really can just go to the whiteboard, come up with a circuit, choose a 6-bit input, and what I'm going to do is grab tiles from the fridge that implement that particular circuit and then run the experiment that day. Okay, and so when I grab those tiles, I mix them together, and what I want to see happening is I want to see in one pot a DNA origami seed form, 6 input bits attached to that, and then DNA strands or tiles attached that grow a tube executing the logic of the circuit shown down in the bottom right corner. So now let me just repeat that in a slightly different way. Okay, so in my fridge, I've got 355 tiles that implement any 6-bit circuit, and I have a bunch of strands that encode inputs. And so what I do is get a test tube, throw in some stuff to make the origami barrel, some DNA strands, and then I choose the tiles that correspond to the program I want to implement, sorting in this case. I add strands that encode the 6-bit input, and then I just mix them together. And then what I do is I heat it up and cool it down to 50 degrees. And we found a wide enough window where we can do seeded growth, a wide enough temperature window. So there was about a 2.5 degree region where we had a large enough kinetic barrier to nucleation, such that what would happen in the test tube is the seed would form first, and then the tiles would bind to that and build the tube. And there was no spurious interaction of strands that are forming structures away from the seed. So we only had seeded growth in our experiment. The data is going to show that later. And this was work with Joy Hoy on establishing this kinetic barrier to nucleation for our 16 helix nanotubes. All right, so after we've formed this cylindrical structure, we send in some strands that actually do a strand displacement operation along the tube and sort of rip it open along its long axis. So now we have a rectangle. We put it on mica, we add some streptavidin protein, and now we see all these little dots. And then if we take an AFM image, we see these structures. Let's zoom in on part of the AFM image. And yeah, so what have we got here? Right, so one thing I did in this experiment, by the way, was I mixed 3 different test tubes together. Each one had a different label on the origami. So 001, 011, 013, and each corresponds to a different 6-bit input shown above it. What we want to see happen is in the orange cartoon on the top here. So we've got that, and then the AFM data below it. So in the first case, we want to see 5 zeros and a 1, and we want to see that 1 pushed to the top. So getting sorted, indeed we see that, and those little circles are the streptavidin proteins, each encoding a 1. And then, yeah, for different 6-bit inputs, we get to see that result. Here's another circuit we implemented, which is PARITY. And PARITY is the problem, given an input bit string, is the number of ones odd. And so our algorithm, our circuit here, what it does, it's kind of cute. It look at this example number 2 here, 002, and we've got a 1, 4 zeros, and a 1. And what they do is the ones run to the middle, try to find each other, and when they do, they annihilate, or they turn into zeros. And if you have an even number of pairs, they just annihilate pairwise, and you get to see just a blank canvas, all zeros to the right. But if there's a single one left over that can't find the other half of a pair, then you just get a stripe written to the right, which answers the parity problem. Here's another circuit. Is the input a multiple of 3? If we were in a room together, I'd ask someone to guess whether this bit string is a multiple of 3 or not, but I'm going to skip that. And here's the circuit run on 4 different inputs. And what this circuit does is it partitions the inputs into 2 sets. if your input is a multiple of 3, it does some thinking for a while, and then it makes 2 horizontal stripes. If your input is not a multiple of 3, like 16 or 53, then it makes this weird zigzag pattern. So this was found by computer search by Eric. So we were at the whiteboard making up circuits, but Eric said, I'm going to try my computer. And then I got really worried because I thought that the computer would outpace me. But anyway, yeah, Eric found some cool circuits with computer search, because the thing on the left, the circuit diagram, is a finite object. So you can use your computer to help hunt for interesting objects. Yeah, cool. As a theoretician, theoreticians, actually, we were interested in what the power of this model was. So if you shove a model of computation in front of me, very quickly I'll start asking questions about, okay, you've got this algorithm, that algorithm, okay, that's fun, but what can it do in general? You know, is it general purpose, for example, or not? And in this case, if we generalize from 6 bits to any number of input bits, which seems a natural generalization, hopefully we can enlarge from 6 to larger input bits in the future. Then if you do that, then you can run any algorithm on this system, or you could simulate any Boolean circuit. And the proof works by simulating a model of computation called Rule 110 that I worked on with Turlock Neary way back in 2006 and was proven universal by Matthew Cook in 2004. And so via this construction, we can say that our model generalized to any number of input bits is computationally universal. So that's kind of cool. We implemented a bunch of randomized algorithms. Here's one that generates cute wave patterns at random intervals. Here's another one that's a bit more sophisticated that Dave Doty came up with, one of the collaborators on the project. And what this does is implement von Neumann's FairCoin trick. I'm not going to explain it in detail, but it's a way to take a biased coin and get a fair yes or no answer out of that coin. So even though your coin is biased, let's say 80% heads, if you flip it a couple of times, if you flip it enough times, you can get an unbiased bit out of it. And yeah, that's a pretty cool algorithm. One thing you could ask is, what is the longest pattern we could make that does not repeat? Us theorists call that a counter. And we found, so by computer search, Eric found a 63 counter, found a whole bunch of them actually. And theoretically, naively, the most you could hope for is 2 to the 6, which is 64 in a model like this. And in fact, Tristan Sterren, a PhD student in our group, showed that there is no 64 counter. So 63 is the best you could do. All right, so in total, we implemented 21 algorithms. And they're sort of flicking up here on the screen. You can read our paper if you want to see more details. There's a lot of different algorithmic behaviors summarized on this slide. Of course, we could. I've just shown you all the cool things we did, but let's analyze it in just slightly more detail. You can see that we have a non-zero error rate. We worked hard to get the errors down. Remember, we spent a lot of time on DNA sequence design. We used that proofreading technique. Yeah, we took lots of AFM images and we analyzed 12,000 structures with a total of about 5 million tile attachments estimated from that. And we found that 1 over 3,000 of those tile attachments were incorrect on average. So that's about 0.03% of an error rate, which is within an order of magnitude for the best in the literature, which actually comes from a system by Rebecca Schulman, our next speaker, and Eric Winfrey, which had significantly less tile types. So we were happy to achieve that. So I think Rebecca got about 1 in 10,000. We get 1 in 3,000 here. So Rebecca's is a little better. The main feature I like about our system is that it's reprogrammable. So it's only got 6 bits, right? So it's not a universal computer, but it's got that property that universal computers have, that you can reuse it for lots of different things. And we characterized it rather well in the sort of theory part of our SI. That reprogrammability meant that we could just kind of program the system while we were at the bench. So that was pretty cool. 40% of our seeds had very little growth off the end, so that's a bit poor. So there's some future work to do there. Okay, cool. There's some raw data. It's just a giant AFM image with lots of little algorithms sitting there on it, executed algorithms, post-execution. You can organize them into a magic carpet, and you can put all your mates on there. So these are the collaborators. I showed this slide earlier. If you want to see a fuller version of this talk where I get into more detail, go to dna.hamilton.ie and search for the word video, or just read our paper. If I have a few minutes, I'd just like to say one or two other things about future directions.
Yeah. I think we're time. Is that right, Guy?
Okay.
Yeah, we're at time. So maybe you can follow this up in the your discussion channel.
Okay, sounds good.
Brilliant. Thank you for your. If that's it, is there any one last thing you want to say to wrap up?
Okay, I'll take 30 seconds. I would like to say that in doing this work, we built on a lot of stuff. We stood on the shoulders of giants, many of whom are named here on the right-hand side. And I think that the techniques that we used and learned more about in this project, like nucleation, error suppression, error measurement, and DNA sequence design, as well as precise control of growth order, could be applied to help us build better DNA tile-based nanostructures. And that's going to be a focus of our group going forward at Maynooth University. And that's it. Thanks.
Thank you.
Okay, thank you, Damien.
All right.